Entrepreneurship and Management under the banner of 5th International Conference on Entrepreneurship, Innovation and Leadership, ICEIL 2023. I'm extremely glad to welcome everyone to the conference under the banner of ICEIL, which was conceptualized in year 2018, looking at the fast-growing and rapidly evolving dynamic global scenario. The use of latest technologies and innovations for the growth of businesses play a pivotal role in the success. The forum intends to bring together the leading researchers, academicians, entrepreneurs, startups, venture capitalists, bankers, innovators, and professionals around the world to a common platform for deliberating with in-depth discussion, sharing of experiences and ideas, and presenting the contemporary research with innovation, best practices, trend technologies, while addressing the challenges and opportunities in businesses. This conference intends to focus on comprehensive research on global targets and endeavors to address the society's greatest challenges in entrepreneurship towards Industry 5.0. The 3 days conference from starting from 15 to 14 to 17, 16 would provide a platform to researchers, innovators, and practitioners to deliberate on advancements on next generation tools that are essential for business. Further, potential entrepreneurship aspirants shall be able to complete a great financial and technical guidance through a proper support which is being provided at MIT University, and the special sessions are being conducted by the experts during the conference so that the entrepreneurs can have a hands-on experiences in form of workshops and keynote sessions, research paper presentations, work symposiums, case studies presentations, which will be held in these 3 days during the conference. And there are various objectives which are fulfilled by this conference to instill an entrepreneurship spirit in the students as well as the community who can be starting their own venture. And they can develop the intellectual self-confidence in form of their independence, decision making, innovative, and entrepreneurship thinking mindset. They can equipped with the required skills and knowledge and experience to be an incredible global leaders, and they can help in the increasing economy for the country. Now, I would like to call our eminent speakers, Dr. B.C. Das sir, Sandeep Arora sir, and are all eminent speakers on the dais for lamp lighting. Girls, please come for the podium. Yeah.
लिखना सिर्फ पोडियम लिखो बताओ इसे काट उसका ये देखिए कल उसको कोई बात है तुम सारे लिखो लिखो जब लिखो पोडियम आराम से लिखो ठीक है चलो
ॐ भूर्भुवस्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् ॐ भूर्भुवस्वः तत्सवितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् ॐ भूर्भुवः स्वः तस्य वितुर्वरेण्यं भर्गो देवस्य धीमहि धियो यो नः प्रचोदयात् ॐ शान्तिः 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 हे शारदे मां हे शारदे मां अज्ञानता से हमें तार दे मां हे शारदे मां हे शारदे मां अज्ञानता से हमें तार दे मां तू स्वर्ग की देवी है संगीत तुझसे हर शब्द तेरा है हर गीत तुझसे तू स्वर्ग की देवी है संगीत तुझसे हर शब्द तेरा है हर गीत तुझसे हम हैं अकेले हम हैं अधूरे तेरी शरण में हमें प्यार दे मां हे शारदे मां हे शारदे मां अज्ञानता से हमें तार दे मां अज्ञानता से हमें तार दे मां अज्ञानता से हमें तार दे मां
Thank you so much, everyone. I would request our dignitaries, Dr. B.C. Das sir, David sir, our eminent speakers to please come on the dais. I would request Dr. Sweta Bawari to bring the Tulsi saplings to welcome our guests. Sandeep Arora sir to welcome Dr. P.C. Das sir. Along with Rupesh sir. I would request Dr. Navid sir and Dr. Sandeep Arora sir to welcome Mr. Sudha Khan sir. Dr. Sudha Khan, I'm so sorry. Dr. Sudha Khan sir with Tulsi sapling. I would request Dr. Nitin Sharma sir to join Dr. Sandeep Arora sir to welcome Dr. Himanshu Babeja sir. I would request Vikesh sir and Sandeep Arora sir to welcome Dr. Navid sir with Tulsi sapling. He is also a deputy dean, so Sandeep Arora sir, please. Thank you so much, everyone. Without further hold up and provide the insights of the workshop, we have with us our respected Dr. Sandeep Arora sir, Director, Amity Institute of Pharmacy. He is a seasoned pharma and academic professional with experience spanning over 27 in pharma operations quality management, academics, training, research, and consultancy, planning and administration. He's a former director Ayush Testing Lab and former funding director Life Sciences Sector Skills Development Council LSSSDC Skill Vigyan Center and is a former president of R&D Natural Solutions Mumbai. He has 3 books, 42 patents, 170 research publications, 4 trademark and 1 registered company to his credit. He has received APTI Punjab Academic Excellence Award as director. He is an academic quality auditor for AQA New Zealand, counterpart of NAC in India, and editorial board member of international publications, including Hindawi UK. So I welcome Pradeep sir for the opening remarks.
The privilege to welcome all the dignitaries on the dais, the external speakers for the workshop on clinical research, innovation in IPR, and the entrepreneurship and management aspects connected with clinical research. So, with the deep sense of thanks to the conference convened by Honorable Vice Chancellor Dr. Balwinder Shukla, with thanks to Honorable Founder President and Honorable Chancellor, and special thanks to Dr. B.C. Das, Vice President STU, as well as Advisor STU, to be present for chairing these sessions. The pre-conference workshop is on clinical research and how it leads into generation of IPR and innovation for the pharmaceutical companies, and how various companies are venturing into entrepreneurship aspects for management of clinical research, as well as providing services for clinical research to the basic organizations, pharmaceutical industry organizations, which are the manufacturing organizations. So, earlier, maybe 15 years or 20 years back, most of the pharma companies, they conducted their own clinical research studies, whether they were for the new molecules, directly going from preclinical to phase 0 and phase 1. And gradually they would go into the next phases, and finally getting NDA approval for coming into the market, and then finally making sure that they were conducting the pharmacovigilance studies later on as the product was introduced to the market. Gradually, in the last 10 to 15 years, it has been realized by the pharma companies, particularly the pharma majors, that having their own clinical research organization costs a lot of much, and it costs extra burden to the pharmaceutical industry. If they are focusing on manufacturing and distribution and providing the appropriate products into the market, naturally having their own clinical research setup gives them with an extra burden and extra cost, wherein they have to maintain all the facilities required for clinical research, which are very different as far as the main aspects of the industry are concerned, that is manufacturing, molecular research, pharmaceutical research, development, as well as quality control and appropriate logistics and distribution. Business development being an important part, because any pharmaceutical industry which requires itself to grow very fast, they need to make sure that they are generating revenues and they need to be taking care of the business. So naturally, it was realized that clinical research has to be a service which can be obtained from other experts, and that is how we have entrepreneurship in clinical research, wherein we have third-party CROs who are providing all the services. When we talk about conducting a clinical research study as a CRO, that means as a CRO, it has to be appropriately planned, it has to be appropriately resourced, whatever materials, whatever supplies and other aspects are required, they have to be generated, and then they have to provide qualitative service to the pharmaceutical industry, the main manufacturing industry. So naturally, they have to act as a service provider to the main industry, pharma industry, and then they have to do it with a target. And the third, another aspect that is concerned with the CRO is that they have to do it in a more scientific and principled way, which are as per the guidelines given by any country. In whichever country they are conducting the clinical research, they need to be following the guidelines. They need to be following the guidelines for appropriate ethical aspects as well as appropriate management of the patients, as well as making sure that there is no harm caused to the concerned volunteers or patients who are registering into the clinical trials, as well as making sure that they are producing the reports which are appropriately statistically vetted, because any clinical trial which may be having whether a smaller number of volunteers or patients or a larger number of volunteers or patients, ultimately the data has to be statistically vetted, and then the significance has to be drawn as to what are the results of the clinical research, and accordingly that has to be given to the main pharmaceutical industry so that they can put it up when they start marketing their product as a validated data, wherein they can show the significance of the studies, wherein they can show the usefulness, efficacy, and safety of the product as compared to the existing products, and thereby they can make sure that they are providing appropriate uh, services and product to the consumers. So, any clinical research operation which is entrepreneurship based ultimately has to act in a different on multiple different dimensions. First of all, it has to be appropriately managed. Secondly, it has to be appropriately scientific. It needs to have appropriate scientific SOPs, standard operating procedures, and then it needs to take care of the needs of the main pharmaceutical industry, which is approaching them as the service seeker, which are called as the sponsors. So pharmaceutical companies are the sponsors, and the clinical research organizations, CROs, they are the actually service providers. So automatically there is a sponsor and service provider relationship that has to be taken care when we are talking about clinical research entrepreneurship. Because of the increasing research, molecular research, deep scientific research, as well as formulation research, product research, as well as the fundamental research, pharmaceutical industry and the new drug discovery process has been happening at a very much faster speed as compared to maybe 20-25 years back. And naturally, it is expected by the society, it is also expected by the regulators, it is also expected by the industry that they should be coming out with not many products in the market with a much faster way. And that is why the need for well managed clinical research and clinical trials, which are producing the desired outcomes in the most statistical manner, genuine outputs, and then making sure that there is no harm caused to the volunteers or the patients who are registering for the clinical trials and clinical research. So that is why we have been seeing a fast increase in CRO. At the present, there are many multinational CROs, there are many middle level CROs as well as smaller level CROs. The revenue generated by the CROs, 75% of the revenue is generated by the multinational CROs. They are occupying 75% of the revenue. The middle levels are occupying around 15%, and the rest 10% revenue is being generated by the smaller level CRO. So whenever the sponsors or the pharmaceutical industry approaches the CRO, they have to first start with initial document, that is the request for proposal, RFP. That RFP, based on that RFP, the CRO starts negotiating with the sponsor as to how the clinical research has to be done, what are the expected outcomes, and what the industry looks forward to when they want to get the clinical trials conducted. Then accordingly, the CRO has to plan other things. They have to develop a contract document. They may be having number of contract documents and sub documents. For example, they may be contracting and they may be subcontracting their services. For example, pathological testing may be subcontracted. Even other services may be subcontracted. The materials for laboratory testing have to be obtained, so they may be subcontracted. So all contracts have to be done. Execution of work is another aspect. Then what will be the takeaways for negotiation with the sponsors? They have to be that. Then there are other aspects also. Whenever there is a CRO, as an entrepreneur, they have to make sure that they have appropriate site management organization. Naturally, any CRO will be having an appropriate site, and they will. April. April. So it's my privilege to welcome Honorable Vice Chancellor Dr. Balvinda Shukla ma'am to be gracious enough to be physically present for the workshop. So as we said, the workshop will be dealing with various aspects for the CRO and the related entrepreneurship. So as we were discussing, when a CRO is developing itself as an entrepreneur and establishing itself as an entrepreneur organization, they need to be constantly interacting with the sponsor or the functional companies for which they need to be doing the clinical trials. They need to take up the request for proposal from the sponsors, then accurately move forward. They need to have a site management organization so that appropriate site is managed for the best management of the patients. Other aspects like they also need to coordinate with sponsors, agent, and other aspects, research investigators. So all these things require planning, diligence, execution, and appropriate FDA approvals, as well as making sure that there is appropriate monitoring for ethics and GCP, that is good clinical practices. So all these things will be part of the workshop. We have experts. We have experts who will be discussing all these things, including primary vendors, secondary vendors for the contract research or the clinical research. So this workshop will have all these sessions covered appropriately by the experts, and we hope that all the aspects will provide the details as to how the clinical research entrepreneurship can be taken forward and how the uh, professionals can further extrapolate into that dimension of pharmaceutical industry, wherein they require support from the other organizations or the third-party organizations, wherein CROs come up and provide support to the industry so that these aspects can be given forward. So I welcome once again all the participants as well as all the dignified speakers as well as the chair and honorable vice chancellor, and we'll be starting with the further sessions.
Thank you so much, sir, for your wonderful opening remarks. It is my profound privilege to welcome our VC man, Honorable PC ma'am. I request Dr. Sandeep Arora sir along with VC Das sir to welcome ma'am with Tulsi sapling. Thank you, sir. Words are less to show our gratitude to our most dynamic and versatile leader, Professor Dr. Balvinda Shukla ma'am, Honorable Vice Chancellor, Amity University, Noida. Professor Dr. Shukla has over 3 decades of rich experience in industry, academia, research, and administration. She is working in a field of entrepreneurship. She has been one of the key team members superheading the exponential growth of Amity Group's engagement in higher education. Under her astute leadership, AUUP has been ranked among the top 3% universities globally by QS and D, which is a very proud moment for Amity University. She is strategist and institution builder, an inspirational leader and quality networker. She has keen areas of interest in promoting entrepreneurship, CSR, quality assurance, industry academia collaborations. She has been awarded with many prestigious awards such as Newsmaker Achiever Award 2014, 100 Most Influential Directors of India Award 2016, Peacemaker Award 2017, Eminent Engineer Award 2017, Outstanding Contribution to Education Award 2017, Women Enabler Award 2018, Phenomenal She Award 2018, Excellence in Entrepreneurship Development Award 2021, Academic Leadership Award 2021, and many more accolades are there in her credit. Thank you so much, ma'am, for your wonderful presence and request for your inspirational thoughts.
A very good afternoon to all present. A very esteemed guest of honor, Dr. J.P. Singh ji, Mr. Suja Khanji, Head QA Pharma, Next Noida, uh, Mr. Naveen Dubeji, VP MCS Live, as well as our very Baveja team and Dr. PC Das, Dr. Arora, Dr. Naved, I can't see here. Yeah, Dr. Naved. First of all, on behalf of Honorable Founder President, Honorable Chancellor, all the deans, deputy deans, head of institution, and my personal behalf, I extend very warm welcome to our distinguished speakers for the pre-conference workshop on the theme Clinical Research, Innovation and IPR, Entrepreneurship and Management during the 5th International Conference on Entrepreneurship, Innovation and Leadership. Um, the objective of organizing the fourth pre-conference workshops were basically to give a very focused insight to the students from the industry, because as we see that the before COVID, and especially if you look at the since the humanity on this earth, we have seen the various revolutions, and the initial industrial revolutions used to be maybe after a century or so, then started shrinking, moved to the couple of decades, and from the by the time the COVID pandemic has come, and I remember in 2017, 18, we were hearing about the Industry 4.0, and MIT University very proudly announced the Education 4.0, where we integrated the technology in all the discipline, all the programs, including the arts, humanities, social sciences also. And then the COVID pandemic came, which brought in many ways everything to stand still, and all people were indoors at homes, no traveling, and many of the sectors were worst hit, and especially if you look at the tourism and aviation was the two sectors were very badly hit due to the COVID pandemic, but at the same time, we had the healthcare sector, which was the savior of the humanity. And I think from the biotechnology, pharma, healthcare sector, when you look at the the vaccine which came, which has saved I would say the humanity and the so many lives, though large number of lives were lost during the COVID pandemic, and I think there's nobody who's not heard or seen either the death in the close ones in the family or the friends or the neighborhood or community. But I think it was the vaccines which have saved the lives so many. To the extent that having the vaccine developed within the country, not only taken care of the Indian citizen, but helped the many of the countries globally, and that to the free of cost. And today, when we look at the global priorities or the national priorities, and healthcare is the topmost, in the chart, where the defense is extremely important, agriculture is very important, space is very important, and environment, water, all kind of things. But I think the healthcare is the prime focus, and we looked at how we can, and it's only about related to healthcare. We have the two workshop. One is which is organized by the MIT Institute of Pharmacy, talking about the clinical research or innovation and IPR, and how entrepreneurship can be promoted in these areas. And secondly, was that how the technology can be integrated for the healthcare, and which was again Dr. Naved and biotechnology jointly they have organized a workshop in the morning. So objective is that all the students you need to understand, as you have the different career progression, and no matter which career progression you go for, whether the higher education or whether you have um corporate career or you have the joining back the family business, but this knowledge which you will gain today during the workshop, it will help in all the careers. It's extremely important. Can these two students, can you get up? This this girl, you and the girl next to you. Yeah, both of you please be out. Both of you be immediately out. Yeah. Yes, both of them. They have something very important to be discussed. They can go and discuss outside, both of you. This girl and the boy next to her. Yes. And note down their name. I will meet them after this. Both of you stand outside. I will meet you people after this session. No, it's important that if a workshop like this is being organized and you people instead of taking notes of anything and talking to each other, do you think that the speakers who have come here, they have plenty of time? You people should take it seriously. Not what impression are you people giving of your institution? So all of you take out your sheets and make the notes. So these workshops are so that you people can understand how the industry is changing over a period of time, what new technological developments are taking place, accordingly what kind of the new skill sets are needed, so that you can take a decision, you know, which opportunity I should avail. Should I start my venture or should I go for higher education or whether I should go for corporate career? And then how do I develop my skills and competencies? You are very fortunate that you are in Amity and so many industry experts, they come to interact with you because they have only one agenda. How can we develop the future talent for the industry who can serve the nation, who can contribute to the economic development? And the economic development only happens when the people are well trained, skilled, innovative, and creative. Because there is so much competition out in the market, and each industry, whether it's a startup or whether it's a big giant, they all need to innovate by understanding what are the societal issues, the problems, and how can we come to those solutions, and then how we can go back to the society with those solutions in the form of products or services of the highest quality at the best possible price. That you can only know when you know your competition well. You can do that only when you know the society well, and then you have the right knowledge, right skills, right competencies, with the thinking mind and a problem solving mind all the time. So you need to have a lot of knowledge for that. And these are the forums, these are the workshops which give you that knowledge. So that your quest for learning enhances manifolds. So I would advise all of you during this workshop, please take notes as many as much possible, And I request Dr. Arora that if you, along with your center heads, if you can also organize a quiz for all these students and see how many have really passed in that quiz, and only give the certificate to participation certification to those students who have passed in the quiz, and all those students who have not passed in the quiz, they need not to get any certificate because they were physically present but mentally they were absent from here. And I want mentally engaged the student who have quest for learning, who have the quest for developing themselves, and that's what the industry will be looking for the talent. When the any expert comes, they also look at the what kind of the students are there. Are they really right kind of the talent to be hired? And this is the moment also to give your impression. And you yourself decide how you are giving the impression about yourself, your institution, and ultimately your university. You know, it's always said that your institution is as good as you make it because your name is associated with all types. So whether you take the name high or you do what you were doing few minutes back. With this, once again, I extend very warm welcome to my esteemed panelists for this workshop, which is very, very important, which has been well conceived and designed by Dr. Arora with the guidance of our dean, Dr. DC Das, and who all the time thinks that how we can bring the best for our students. And the morning workshop, again, I saw what Dr. Naved and Dr. Kamal Rawal has designed on the bioinformatics again was very highly designed workshop, and seminar hall was jam packed, and more students were flowing into the workshop because they wanted to learn. So therefore, please develop your learning attitude for all, and the lifelong learning is one of the most important attribute for everyone. Well, even at my level, I need to learn every day that what is new is happening, what is new is coming, so that how we can give the best possible curriculum to our students, best possible support to our students for developing them the industry ready. So wish you all the best, and my best wishes to the success of the workshop. And I wanted to come when the lab lighting was happening, but we had at that time the interviews of the faculty positions where I didn't want them to wait for me. So I said if I have the time, I will come. And personally, I just so that I will continue to see the workshop online, and also I will deliver my address. But I'm very happy to be here with all of you and meet all of you, especially our distinguished speaker for this workshop. So thank you very much.
Thank you so much, ma'am, for your inspiration always, and your presence always motivate us more. And we are also very keen to always listen to you. And that's an irony that students, you are not extracting out the information from the sessions. Please be very attentive, as ma'am has told. These are going to make you ready for the industry. So please listen to the sessions very, very carefully. Um, Now I would request Dr. Sandeep Arora sir, along with Das sir, to welcome our honorable guest of honor, Dr. Jayakash Singh, Senior Principal Scientific Officer, IPC, and he is officer in charge, Pharma Covigilance Program of India. Sorry, sir, for little delay in the welcome. We are blessed to have you with us, sir. Now, to give a deep knowledge about clinical research and innovation, we have a person with ocean full of scientific and research knowledge. Our Dean, Health and Allied Sciences, Dr. B.C. Das, sir, Chairman and Hargobind Khurana, Chairperson, Amity Institute of Molecular Medicine and Stem Cell Research, Amity University, Uttar Pradesh. Formerly Founder Director, Institute of Cytology and Preventive Oncology, ICMR, Noida, ICMR Delhi. Dr. Das had made outstanding contributions in the field of cancer research, tumor virology, human genetics, and mutation research. He worked several years with Nobel laureate Professor Harald zur Posen, Nobel Prize 2008, at German Cancer Research Center, Heidelberg, Germany. He is a recipient of Dr. B.C. Roy National Award, Sandoz Oration Award, Ranbaxy Medical Research Award, J.C. Bose National Fellowship from 2008-2016, and he has guided many PhD students. Number is 38 or more, and 79 MD students. Sir, we would look forward to listen to you.
Thank you, Professor Amanvalia. And very good afternoon to one and all the dignitaries of the dais, specifically Dr. J.P. Singh, or the chief guest today, the guest of honor. And there are several, I do not know how many are participating, but those who are present here, I see Dr. Suja Khan, Mr. Suja Khan, and he is from QA Pharma Lex, Noida, and Dr. Himanshu Maveja, and he is also from the same organization, and of course the others are there. They might. Huh? I am not audible. Is it audible now? Yes, it is possible. My voice is very loud, but because of last 10 days, I was down with fever and all. So that's why I have just first day today joined here. So that is why it is the voice is a bit low. So now it is audible? No? No, there is some problem. So, is it not?
Hello.
हेलो हेलो नहीं यहाँ पे प्रॉब्लम है कहाँ है हेलो हेलो is it audible?
Yes, sir.
Yeah. Now it is audible. Okay. So so there's the other the panelists may join later on, like Dr. Pooja Sharma, whom I know personally. She is from Vedanta Institutes of Clinical Research in Gurgaon. And there are, I think, Naveen Dubey, and there may be another persons there, but they may join later. But at the outset, on behalf of our Honorable Founder President, Dr. Ashok Chauhan, and the Honorable Chancellor, Dr. Atul Chauhan, and of course, Madam Vice Chancellor, Professor Dr. Balvinder Shukla, who is the chairman of the 5th International Conference on Entrepreneurship, Innovations and Leadership. So this is ICEIL, and this is a 5th international meeting. I think what he was telling that all the students, those who are listening to it or participating to this workshop, if they are in the first year, second year, if you will be in 4 years here, at least, and those who will do the M.Pharm later, they can have 5-6 years together, because every year it happens here and in a very big way. And you will see how Mela it becomes tomorrow onwards when the inauguration is done, how many sessions and how many people, how many industry, and how this activity, it's really a great activity. The whole campus is involved for this till weekend. You will see that, and if you can, and there is no end of learning things. And what she was telling is you have the ample opportunity to learn this time. And once you make your career right, you can enjoy endlessly. There is time to enjoy, and there is time to learn. This is the time to learn. And I am sorry, or I am really little happy that I also lost this temper in the other meeting in the last week, that the physiotherapy day, in the same on last Wednesday it happened, same, and around this time, and it was such that I was addressing and few students were gossiping, and so I lost my temper as Madam Vice Chancellor did. I said, "You get out, and if you are not interested, I am not interested to talk with you or asked you to listen, and you better go out. Don't disturb others." So this is the right thing. So this is what, if you have come, if you don't want to come, don't come. Nobody forces you. And if you are really interested to come, then come and listen to it. So this is the most important part. So, so this is the, and of course, I welcome you all and all the dignitaries on behalf of the authority of the MIT, as well as on my own behalf, and all the faculty and the students of the MIT Institute of Pharmacy, and all on behalf of the MIT the Health and Allied Sciences as a dean. So this is an excellent opportunity to learn, and there are three buzzwords, buzzwords that is entrepreneurship, innovation, and leadership. These are the three important things we have understood. Now it is just immediately it is the G20 closed, and we have the repercussion in the campus because we had the S20, Science 20. The part of the G20 was held here in the last week, and so just before 3 days before the G20 starts. So this is, we know, and you are all witness that, because that was held in the working day. So this is what is most important, that how the industry and economics and entrepreneurships and everywhere it is innovation and the IT and what is not, and the artificial intelligence and entrepreneurship. Most important is entrepreneurships. But the route to the entrepreneurship, and there are many routes. And as Madam Vice Chancellor has mentioned, that in a society or in a country or in a nation has to progress only when it's not only education and economics and the technology and other things, but most important thing, which is mostly neglected in our country, is the health. So it is a good thought out or thoughtful decision to have the what is the role of the health and how the entrepreneurships can be initiated in the field of the health and allied sciences or health sciences or biomedical sciences. And one of the area is the pre-conference workshop which is being organized in the clinical research, innovation, and IPR. Three are more important because these are the source of the entrepreneurship and the management, and rather to discovery of any new drugs or any in the field of, if you want to contribute something substantially in the field of the health sciences or the healthcare. So this is the area, one of the area, but then there are pharmaceutical sciences, as you know, it's a mother science, and it has a ample opportunity to start from any disease to being cancer person, to cancer, to diabetes, to mental health. any type of disease, any type of drug discovery, any type of pharmacophore, any type of formulations, everything can be done here with this discipline. But then to get the excellence or to reach to the entrepreneurship or to make a mark in this field, you have to go through clinical trial. So this is one of the aspects what clinical trial, how much you have the knowledge, I do not know because I teach clinical trial in the biomedical sciences and I have my special interest in this area and I teach also the ethics to the PhD students and all, so which is close to my heart because ethics is everywhere in your life and this clinical trial is the first thing is that there are three, 4 phases, phase 1, phase 2, phase 3 and preclinical trials or preclinical studies or which is laboratory based. Then you go to phase 1 and then phase 2 with a small number of individuals and then phase 2 and then phase 3. And that is the thing. Phase 4 is the market study and all these things. But till phase 3 is the major study is done. And you know this thing you have heard, you must have come across the COVID vaccines and how it was emergency introduction after just having done this phase 2, even not completed phase 2. So it was introduced, this vaccine because of the emergency, the preliminary studies which indicated with the small number of samples or the cases or the human samples or human volunteers. So this is the route to establish your discovery and then you make it patenting or IPR and then you can have these molecules because before this any molecule or new drugs come into the shelf of the pharmacy, before that it has to go through all these aspects, particularly the clinical trial. And clinical trial involves all types of preclinical studies and clinical trial and where ethics and everything and all the 4 stages, everything, ADME, SAE, ASE, everything, all adverse effects and everything is taken care of and ethics is also taken care of. And most important point in the ethical, the clinical trial is that is ethics, that you cannot have any the cruelty to the individual, the living organisms. Means if it is mostly the clinical trial in response to human trial, and so you cannot exact any cruelty to the human subjects either through any treatment or the and if it is a clinical trial, the animal trial, you have to also get through the animal ethics clearance and human ethics clearance. So I have been half a dozen committees, including Delhi University and this hospital, the Sarita Bihar, that what is that hospital? The Sarita Apollo. So I am in their ethical committees. So so it depends because my area is on cancer and stem cells. So mostly I am in the stem cell ethics, but Delhi University I am in the human ethics. So this weekend is the meeting. So there are a lot of clinical trials. There is less clinical trial, more of the other studies. But the medical related hospitals and pharmaceutical organizations and industry, they are with the clinical trial. I will not go into the details of that CROs. What is the responsibility of CROs? Certainly CROs have a lot of, it's a very complicated area rather because it is highly expensive. And but whatever it may be, you will come to know what is CRO and how the clinical trial is conducted, because you yourself cannot be the PI of the project. So it is to be given to the third party to do the conduct the clinical trial, and where all the ethicals, all the aspects of the to be taken care of, including the ultimately the dose and dose response and everything has to be, and there the bioavailability and all studies are done. So ultimately, if it fulfills everything, then the CGI is satisfied with that. Before that, the CGI has to be given the permission to carry out the clinical trial. And for that, you have to do preliminary studies to submit the data, and then the DCGI the Director General of the DCGI, Drug Controller General of India. So they will permit to continue carry out the clinical trial, and then you submit all the data of the clinical trial, and then they will approve it that yes, this drug can go for a human use, and then only you will find the drug. If a drug is started, you may start with the 100 molecules, but you can left with one molecule, and it takes 15 to 20 years to see that molecule on the pharmacy shelf for the human use. So you can think that how long it takes. So there are several methods, that the methodology, the resource management, and then the clinical research innovations and entrepreneurship. So it is this conference or this pre-conference and this associated with this the ICEIL, that is the entrepreneurships and innovation and leadership meeting, international meeting. It is attached to this to see that how entrepreneurships can be attached with this health sciences or healthcare aspects for pharmaceuticals and all types of pharmacophore and all types of derivatives and type of the isolations, phytochemical isolations and all other. So, but these are the preliminary step, what you do during MPharm and all these, the thesis and all. Those who are MPharm students, they will know. Those who are PhD students, they know better. So it takes a lot of time, and it is important to know how it can reach to the ultimately you declare this molecule is fit for human use. So that is nothing but at the state of the drug or the medicine, which can be consumed by which is not unsafe, it is safe to use. The dose and all these things are prescribed, and then only it is given to the human. So this is the safety of the human or the humanity is very important before you do the clinical trial. So these are the basic aspects of the clinical trial. Just I wanted to tell you, and to have those you may not have that idea. So that's why I just briefly explained. But certainly how it is the mental your thinking is to be that anybody can do this thing, research and discover drugs, but how do you can go for entrepreneurships or the startups you can start. So there are several examples. So I will not go into the details. So I would end it here. So there are several experts from the industry related and from the pharma company. So they will give the more indications that how you can enter into the entrepreneurships and patenting and all IPR and all. Of course, anything you can in your initial stages when you have thought of, you can design this and you can have the patents done. For example, when I joined in the beginning in the 2014 year from the university year, I had thought of designing a molecule which is called triple conjugate against the drug resistance, the cancer stem cells. So there is no drug till date today. Even there is no drug to control how to overcome the drug resistance or how to kill the cancer stem cells. These are the cells which are refractory to the radiation and chemotherapy and all the other therapies because they produce some protein, the transmembrane protein. That protein has a capacity to reflux out the cytotoxic drug. Any cytotoxic cancer drug is given. The 99% of the cancer cells are differentiated cells. They are killed by the radiation or chemotherapy cytotoxic drugs. But these are 1 to 2% cells. They cannot be killed by any cytotoxic drugs or radiation. So they have the capacity to reflux out the drugs. So that's why. So what we did is that we developed a triple conjugate, and that time I just concept and had the patent filed. And after 4 years, it is just 2 months, 3 months before I got the approval after 4 years. 4 years, no, 5 years, 6 years. So now it is in the clinical trial, but the animal trial rather. So then it will go to the clinical trial so that you do that. What is that? Just to 1 second, I will say that there are 3 components of it. you have one molecule to target to the cancer stem cells. So that is the targeting ligand. So once you inject, it will go to that cells only, not to the normal cells, not to the other cells. So it will go to the cancer stem cells. It has a, because it has a high amount of the folate receptor to bind. So this is folate receptor, the targeting ligand, and it will bind there, and then through endosome formations and the Catherine coated pits, it will be taken in, and it will go to the cytosol, and the pH of the cytosol will dissolve the bonds. That is, three molecules are joined together with the help of the synthetic chemist. So that bonds will separate out the drug as well as the another molecule, which is the phytoproducts. So that phytoproducts have not been independently studied, and we have proved that this phytoproduct can sensitize the cancer stem cells. So the cancer stem cells is now get into this molecule, and this will now separate it out and sensitize the cancer stem cells. And now the drug, the standard drug, for example, doxorubicin or any other drug, it can easily kill the cancer cells. Otherwise, it will not be able to kill. So this is the basic principle, and which has worked so far. The preclinical and other study and an animal study is going on. So hopefully we will be able to, but it will take another few years to go to do the clinical trial. So as I told, it takes 15 to 20 years to get a molecule done. So this is something just to inspire you, and to how exciting is the field, that you can know that one drug can change the, like Taxol. It was a plant product, and it changed the whole picture of treatment of many of the cancers. So there are ample opportunity to have discovery some molecules or some entrepreneurship. One can do wonders, and you are the 21st century young, and you have the ample 80 years, 90 years. You are maybe 20 years, because 100 years, if I take minimum age expectancy, life expectancy, you must have completed only 20 years. So 80 years is still at your disposal when you are intelligent enough to decide how to do and how to go about it. When there are a huge line of aspects of the area, you can go and you can do wonders if you want only. So you have to be focused in your career. So I wish all the best for this workshop, as well as to all the organizers and to the students. Mostly, you are the most important users. So this is not to just to lose any temper, or just Madam Vice Chancellor told, but to take it sportingly and to utilize the best utility of the knowledge. Knowledge is yours. Nobody can take it. Everything can be taken from you by anybody. But knowledge, only when you die or we die, then this knowledge ends. But knowledge differs from person to person. No two persons are similar genetically. But no one is less than general capacity. So you should try your best, what you can, excellence in your career, wherever you want. All the best. Thank you.
Thank you, sir, for your valuable insights on the theme. And very beautifully, you have explained about entrepreneurship, and actually, which cannot be achieved by doing innovations. And sir, these terms are really new. Innovation and entrepreneurships are really very new. But you actually started doing innovations in the cancer stem cell long back. That is too inspiring. So students, you can actually be taking a lot of knowledge from Das sir, because he has worked in innovation field long back when these terms were actually not there. So without delay, then as our one speaker has joined online, so I would request, before we join with Dr. Naveen Dubey sir, I would request Dr. Sandeep Arora sir and Dr. Suza Khan to felicitate our Dr. PC Das sir. Thank you, sir. Now, I would request our next speaker, Dr. Naveen Dubey, sir, Vice President Operations Medicine Service, Lucknow. He has joined online because of some prior assignments. Dr. Naveen has 20 years of experience in the fields of analytical development laboratory and bioanalytical laboratory. He has a Doctor of Philosophy in Chemistry with thorough academic knowledge and understanding of pharmaceuticals. Creative, ambitious with strong analytical abilities, excellent research skills, and strong managerial abilities. He has worked on drug and drug product analysis during development, optimization, and stability products of ANDA, NDA, and IND applications in different organizations like Sun Pharma, Ranbaxy, and Jubilant. He has worked as an associate director, bioanalytical operations, Jubilant Generics Limited, with a team size of 24 scientists. Department is involved. He is involved in the bioanalytics of generic and NCEs molecule meant for human animal trials through his sophisticated through using high sophisticated instruments like LCMS. I welcome you, sir.
Hello, ma'am. Am I audible?
Yes, sir, you are audible.
Yeah. Thank you, ma'am, for nice introduction. I thank you, Dr. Sandeep and all team. Actually, I was supposed to come there, but due to some business priority, I could not attend physically there. But due to his request, I am doing it from live. I am actually in the car, but I will do it through this car only, and for the benefit of the student and other audience. So I am sharing my PPT. Is it visible to you?
Yes, sir.
Yeah.
Yes, visible.
Okay, sir. Okay. So I'll start like the topic is entrepreneurship in the clinical search. So I'll start. I have selected some points on it, and it will be beneficial for the student and for all the team members here. So by terminology, what is entrepreneurship? It is the ability to develop, organize, and run a business. So like we can think of two kind of scenario here. Like one scenario where the business is from say like from grandfather, parents, and current generation. Like I can give you an example. Like Sun Pharma is the one of the example where the pharmaceutical business started from their grandparents, and now Dilip Sangvi is heading it, and now his offspring like son is also heading it for the country. Other example is Jubilant. Jubilant Bhartiya Group it is actually from say, their grandparents to the Bhartiya Group, and now their uh, children are heading it. So, but all of us, like most of us, cannot have this kind of privilege. So we need to learn it, feel it, and then we can become a entrepreneur. So I'll start from the history of this clinical search in the field of pharmaceutical. Like this is an example of thalidomide. This was a drug which was developed in 1957 and it was withdrawn in 1961. Only 3 year or 4 years time span, the drug is launched and is withdrawn from the market. What for it was launched? This was launched for morning sickness of the pregnant woman, but it was causing birth defect, body deformation, and ultimately the regulator has to stop this drug. And why it was stopped, why it was withdrawn? Because the clinical search that was to be done during the development phase was not performed. What they did, they did some clinical trial, but they did not consider pregnant women. They did not consider toxicity effect, and they did not do the animal trial also. So after the regulator ask them to withdraw this drug, they again gone back to the laboratory. They did animal trial. They did involve some pregnant women also, and they found that this drug is not safe in pregnant women, and toxicity is very high. So after some modification, they came out with this drug again in 1998, and now this drug is used for leprosy, multiple myeloma. Means the drug designed for something is used for something. So they have to do lot of R&D. The another example is rofecoxib is the drug launched by Merck in 1999, and it was also withdrawn from the market in 2004. So within a short lifespan, the drug is out of the market. So why it was out of the market? Because the drug is meant for painkiller, but it is causing heart attack to the patients. So we need to understand during the business point of view what we have to do, why we have to do, and how it will work. Otherwise, the problem will be there. You have to invest $1 billion, and ultimately you will come into the loss. So we need to understand the laboratory work, the documentation work, the regulatory aspect, and the human aspect, ethical human aspects. So we will start like we'll try to understand what are the drug development process. The drug development process can be divided into mainly 4 steps. Initial phase is drug discovery, then preclinical, that is done in the animal study, then clinical search, and then the approval processes. So we'll start from the drug development. Like before the drug development, we need to understand the business. Business means what is the type of disease that is there in the market, and what is the revenue that we will receive from the market. Like there are many drugs, like there are many diseases which will be boom for the market. Like cancer, you think of cancer, there has to be some drug, and it will be like non-ending. You will get lot of revenue for it. You think of diabetic, you think of BP. These are the current existing diseases, and there are a lot of research going on. There are a lot of medicine available currently, and still there are thousands of studies going on this kind of disease. But think the other way, the disease like Alzheimer, the disease like cerebral atrophy, they are there in the globe, but the patient pool is very low. So the businessman, like the person who are going to invest in the business, will think many times before going for the drug development of this kind of disease, because the patient pool is very less, the investment is very huge. So they will not think of going for this R&D. The currently few months back, there is a drug, or in today news also, there is a drug which is costing 16 crore, 10 crore value, spinal muscular atrophy. So one normal person or one even the rich person cannot afford this kind of drug. So what they need the funding, government funding, public funding to have this kind of drug. The company investment is very huge, and the reach on these drug is very low because of the costing. So unless there is a number of pools of the disease or the person or the volunteers there, the company cannot think of. Why cannot think of? Because they have to invest in the R&D. They have to think of the patient pool. Then they have to think of the time they will spend on the research. Who will fund it? Government is funding it or not, or they are pool of the funders, or they are self-funding it. And the time regulator will take to approve this drug, and then the how much time they will get patents. So these are all factors one need to think of before going into pharmaceutical business. So in pre-development, like the preclinical study, there are discovery phase, there are animal study phase. Within the animal study phase, there are a lot of testing is there, like pharmacological testing, hypersensitivity testing, genotoxicity, carcinogenicity. So a lot of varieties there for the particular drug, whether it is oral, dermal, or something else. So based on their variety, that animal study can be designed. Within the drug discovery, like it can be done conventional way or the computational way. So currently, like in the corona period, we have seen the computational way. Conventional way will lead to say 10 year, 15 year for one drug to come into the market, but using the computational way, we can think of coming a drug into market, say within 2 to 3 or maximum within 5 years, we can launch a drug into the market. How we can do it? Think of chemistry way. We can design, we can think of the disease, we can think of using the software kind of molecule we need, and the computer do all the work for you. Like they will think of hydrogen bonding, force interaction. They will think of how the compound is interacting with low energy, like the molecule is very into the active side without any problem. So they will think of this and they will propose some structures. And after proposing their structures, we can think of synthesizing that into the lab. So how many software? There are a lot of software that can help for development of this kind of molecules in the lab. So some of the example are Dalton's. You can think of Forge. These are some of the example you can think of. You can search. Lot of software are there which are there to help us. But we need to think from the businessman point of view, like which software will we have to buy, and that software has to be FDA 21 Part 11 compliant software, so that one, if the auditors is coming and they will see that how you are doing the homework, so you can show all the trails, like which molecule you have selected, which molecule you do not have selected, and why you have selected. So all trails should be there in the softwares. Starting from the animal, say passing from animal to human, we need to do like we have to test to the human. So there are some example for this. Clear. We have to learn from the past our errors. In initial way, like one antibody or hexamethonium or the gene therapy. In these molecules, the companies have directly involved more than one patient. So what was the side effect of this or after effect of this? For the antibody test, they have involved, it is a study in the UK. They have involved 6 volunteers to human while switching from animal to human. So what they found that all 6 volunteers died on the spot within the phase 0 trial. So our aim is to see the safety first in the human. When it comes from the dosing is very, very low, say 100 times or 1000 times, we are going low dosing, low dose. So we have to think on this, whether it is 100 times down or 1000 times down, whether it is safe in human or not. So we have to select only one volunteer, single volunteer. But the error they did in the past, like UK and US, they have done they have involved multiple volunteers, and what they have seen, most of the casualty they have seen. So after that, the regulators recommended to the pharmaceutical company that you cannot add more than one volunteer for phase 0 study. You need to see the safety of the drug first, whether it is safe in human or not. So this was the aim in the phase 0. And then the phase 1 comes. What we see in the phase 1 trial, we see the safety, like the if a dose is to be made, say 50 mg. So we need to start from 1 mg, 15 mg. We have to test whether we have to make the cohorts. Like some group will be given say 1 mg, some group will be given 5 mg, 10 mg, 15 mg. So we need to compare all the results. We need to see the pharmacokinetic of that those all cohorts, and viability, bioequivalence, metabolism, toxicity. All these data has to be compared for the different formulation of the same molecule. We need to see also the maximum dose tolerability in the human in the phase 1. Then in the phase 2, what we have thought while making the protocol, like this molecule will work for this particular disease. This is called proof of concept. Here what we see the results of the phase 1 study, and we see that what is the minimum dose that is causing the therapeutic effect. Say in phase 1 we have tried many formulation like 1 mg, 5 mg, 10 mg, 15 mg, 20 mg till 50 mg. So whether it is 1 mg, whether it is 50 mg, we have to see the result from the phase 1, and then we have to extend it to the multiple volunteers. We see here the minimum dose, effective minimum dose for the volunteer. And this is the main criteria from here, we move to the phase 3 and phase 4. So we see that we have to fix here the particular dose, and we cannot move more than like multiple dose from here. In phase 3, we increase the sites, we increase the volunteers, and what we see here, we see the efficacy of the existing dose, like existing molecule. We do blinding, we do non-blinding blinding, multiple site, and we have to see whether it is causing benefit to the larger population or not in the phase 3. And then phase 4 comes. Like after phase 3, we file it to FDA. FDA just grant the approval, and after approval, it comes into the market. Now, say we might test it in say 10,000 volunteer, 20,000 volunteer, but once it comes into the market, the number of people who use this molecule may be lakhs, 2 lakhs, 100 lakhs. So number of volunteers will be very huge. So what regulators say that you have to see the after effect of the say drug post marketing, like whether it is causing, whether it is safe or not, whether it is causing what is meant for it, whether it is acting similar way or not. Like example of the thalidomide and the ropicoxib, they were banned after the drug is launched into the market. So after within the phase 4 trial, they have been like banned from the market. So this is the main like the main we can say criteria where the actual drug testing is done. It is done through the hospitals, through the OPDs. So a lot of people are involved in this to evaluate the actual efficacy of the drug. So in summary, we can say that the phase 2 trial, 3 and 4 trial is meant for say human pharmacology, therapeutic exploratory, and then therapeutic confirmatory test, and the actual therapeutic use. So these trials are very huge trials and manually cannot be done. So there are many software that has to be controlled, like say multi-centered, we are doing the clinical trial. So there are software who manage it, like the doctor, the patient, the CRA, who are doing in one side, other side, multiple sites. So they parallelly enter into the software and the data is recorded, and the person who is reviewing the data, the QA department, the QC department, they just evaluate this data, and then all the data is compiled through the software, and it is easy to use for the regulator to assess the assessment of the trial. So once the understanding of the clinical trial is done, being an entrepreneur, you need to think of the how to do and where to do. Like you need to have some organization where you can conduct it. You need to have the people who are managing it. So it is a basic requirement that once you are thinking of setting up a business module, you have to have a legal identity. Like you have to have a particular area space where you are conducting your trial, your research, and you need to have the qualified who are managing these trials. Like you need all legal approvals for your fire, water, safety, pollution, and all those things that will be required for a center to run. The person involved in this organization to conduct the trial, any kind of study, any kind of research, has to be legally qualified. If you need a doctor, you need to have at least minimum graduate doctor, or you need to have minimum graduate scientist or pharmacist. You cannot have any person and you can hire them for doing actually work. That is actually not valid. If you hire some commerce people to do science or art people to do science work, it will not be beneficial for your research work because they are not trained for that kind of work. So you have right people to do right research, and then you need to have in the department, in the facility, you need to have proper reagents, proper chemical, proper equipment. The calibrated equipment is required. So each thing available in your facility has to be properly justified, properly guided by the work instructions, SOPs, and how to use it, how to discard it. All those things has to be there in your department. The SOP play a very vital role here, like the for each processes, like whatever you are doing in a trial, who is doing in a trial, the role and responsibility of the people, the calibration of the equipments, like the usage of the chemicals and the proper safety procedures. All those things has to be written in a SOP, and it is readily available to everybody, like the person who is coming for. So each equipment must have adjacent SOP, so that it is ready used for the reference purpose. Even the regulators comes, they will see it. Some equipment is there, some work instruction is there, so they will review it. They will see that you are following proper GLP guidelines here. There has to be some contract and agreement with the vendors. Actually, all the things, all the work you cannot do alone. Means we have to involve multiple team members, like the vendors, the equipment vendors, the service vendors, the hospitals, and the project management team. So multiple things are there which are involved during your work, clinical trial or any kind of study. So we need to deal with the confidentiality agreement, who will do what, and to what extent they can think of, like they will come to know once they are there with in your organization, they will come to know of your activity, and they can reveal it to somewhere else. So before taking any kind of work from them, you need to have an NDA signed with them, so that they cannot reveal to third party. Means between you and them, it is fine, but any third person should not know what you two are doing it, and what level of service agreement, what he will do or she will do, has to be written properly in an agreement, and the annual month maintenance contract has to be there for all the equipments which is being used in your organization. And this is the very crucial department, quality control and quality assurance department. These are the department who will check, who will monitor, who will control the your accuracy of the data, your ethical conduct of the data trial, and they are responsible for all kind of audit, future audits. So these department has to have a good number of people in the organization. The QC department is daily, regularly involved in doing the procedural check of your work. Maybe it is 100% also. Whatever the team is doing, they will cross check it, and they will evaluate it, and they will justify it after the trial is being done. And the quality team, like quality assurance team, is there to assure that the data, the trials is being done as per the GCP GLP guidelines. And after the data trial is complete, there will be a lot of data there in the organization. So we need to have say one safe house, which is known as archival facility, which is to be like temperature controlled and humidity control environment. It has to be in this environment, and it should be fireproof environment, so that because the data we keep here is meant for say minimum 5 year to maximum 25, or even for the lifetime also. So we need to have one archival facility in the organization which will keep the data ready in the facility, and this will be used say for any kind of audit. The regulators come say after 2 year, 5 year, 10 year, the data is there, and you can show it to them by retrieving from this center. So this is all about say small entrepreneur. So once you are thinking of say setting up this kind of business, you need to first feel it, learn it, and then you can think of putting such kind of say clinical trial business. Thank you.
Thank you so much, sir, for your valuable time by joining online despite of your busy schedule. Thank you so much for enhancing the knowledge of participants in the field of clinical trials, which is very significant part in healthcare. The students can achieve success by following the SOPs, which is a very important part of the industry, healthcare industry. And we have to be doing all the things in a thorough manner by using and following those SOPs, which was actually beautifully explained by you. And the record management, which is again one of the very pivotal part. Whatever we do in industry, the record maintenance is also very important. So that has been summed up by you in a nice manner. Thank you so much, sir, for your valuable insights on the topic. Now,
Thank you.
So thanks for very good for taking off.
Now, our next speaker and our guest of honor, Dr. J.P. Singh, sir, Senior Principal Scientist and in charge, Pharmacovigilance Program of India. Dr. J.P. Singh, sir, has his alma mater from College of Pharmacy, presently DIPSAR, New Delhi, and he is doctorate from Department of Pharmacology, AIMS. At present, he is a senior principal scientific officer and officer in charge in Pharmacovigilance Program of India at IPC Ghaziabad. He was also secretary cum scientific director IPC. He has overall more than 20 years of research experience in the research and teaching experience apart from pharmacovigilance and formulary science. He was temporary advisor expert for WHO's expert committee Geneva, Switzerland. He was a member of Institutional Human Ethics Committee, National Institutes of Biological Noida, member of Core Group Expert Committee for revision of National List of Essential Medicine 2011, 2015 and 2022. He is a member of CIDESCO Expert Committee on DRACE test. He is a member of National Expert Committee for Allergens and member of International Society of Pharmacovigilance. He is also a member of Multidisciplinary Expert Committee of National Pharmaceutical Pricing Authority, NPPA, New Delhi. He has made several presentations in national and international conferences. He has many national and international publications and book chapters into his credit. Prior to joining as principal scientific officer, he served as principal scientific officer and senior scientific officer in IPC, grade 1 in Central Institute Pharmacopoeia Laboratory. He worked as a lecturer under the Directorate of Training and Technical Education, Government of India. He has several publications, 16 book chapters, national and international publications, and he is a recipient of Miss Geeta Mittal Medal for Basic Research in Field of Oncology for being the best postgraduate at AIMS. Servicer Young Investigator Award and Certificate of Merit in Pharmacy. So very big CV, but I have to read few of the things in it. Thank you so much, sir, for your valuable presence.
Vice Chancellor Madam, Dr. Balvinder Shukla, Dr. D.P. Dr. Das, Dean of this institute, all the respected dignitaries, my colleagues, Dr. Navneet Sharma, Dr. Javed, Dr. Puneet, the galaxy of the faculty and students sitting here. I feel really honored to be the part of this important international event on entrepreneurship, innovation, and leadership. First of all, I congratulate the organizers for inviting me to this important conference and the youngsters who are the student of this institute. They are in the best hands of the faculty
Last lecture to make sure that
who are
not to be repeated again already.
who are well versed with the discipline of pharmaceutical science. And pharmaceutical science, it is a very vast science which also involves the clinical research. And the previous speakers, they have also threw the light on the innovations, what is entrepreneurship and how should we go ahead. So I need not to repeat all that, but I think the first thing to be innovative is that you have to be
you have to have focused attention, and you have to decide your goal, what you want to do in life. And for that, you also need to maintain lot of patience, and this you will learn right from the beginning. And I think many of you who have come to this institute, even if they were not aware of the environment, then by coming in contact with the learned faculty of the institute, they will also try to change. And the innovation and the innovation is will also not come just in a single day, and you have to be dynamic, you have to think about it, and it should be a new thinking, then only it will be acceptable. Your patent will be accepted only if there is something new which has not been invented earlier, and then only you will get the right of exclusivity either in our country or in any other country, but you have to file the proper application there. And for this, you have to continuously do the research, and your research starts from the PG. Like when you are in first year of master's degree in pharmacy, you are given the demonstration, you also do the experiments, and then in the final year, you are given the exposure to carry out the research. And as it was just told, having the SOP, so one has also to have the SOP for carrying out the research. And if the research work is not properly documented and properly published, it will not be accepted, and it will also not come to the notice of other researchers. And for that, you have also to think of the type of the journal where you want to send your research for publication, whether it is in your field or it is out of your field. So if it is out of the scope of the journal, then outright the editor may reject it. But if they find a substantial thing in that, they will send to the peer reviewers, and after examination by the expert in that particular discipline, the editor will get back to you whether your research can be accepted as such, whether it requires minor revision or it requires extensive revision, and they will also suggest you the further line. If they reject it, sometimes they will suggest some other journals where you can send your research. And for this, you require the proper designing of the protocol for the research, proper role of the type of the scientist, like the which method of statistics you will apply at the end of the experiment. And if there is no statistical power in your research, then there are chances that your research will be rejected, isn't it? And therefore you have to continuously do the effort, and until and unless you are monitored by your boss, by your supervisor or guide, then the improvement will not take place. The improvement will take place at a greater pace if you are more frequently interacting and discussing the protocol of your research with your guide who is well versed in the subject and will make your study protocol or research easier, and you will be able to accomplish it in a time bound manner. Otherwise, as you have been given a defined timeline for doing the research, and if you are delaying, then you can think of what would be the repercussions, and you may go into failure also. And for this, it requires lot of interest in the subject. Alone, it will be difficult for you at the initial stage to carry out the research, and your mentors will help you a lot for the research, right? And therefore, you must continuously be in touch with the scientific literature and the systematic search, and then to decide what is the lacunae in the current study which we can take forward to fill the gap. If you if you invent anything new, then you can file a patent in a relevant patent office. And particularly in the area of the pharmaceutical sector, particularly the new drugs, they require rigorous scrutiny as well as research. As you have witnessed that many of the drugs formulations which were approved, they have been withdrawn from the market, and this has been possible because of the evidence based outcomes on the safety of that particular product. And earlier we were depending on the Western data, but now there are a lot of changes. Despite the challenges, that today we can take the decision on the safety matters through the available evidence on the Indian patients, and that's why you may be knowing the name of the Pharmacovigilance Program of India. And Government of India, Ministry of Health and Family Welfare has shifted the National Coordination Center for Pharmacovigilance Program of India to Indian Pharmacopoeia Commission around 12 years back. And since then, we have tried to expand the Pharmacovigilance Program of India in different states of the country. And to my knowledge, there were around 22 adverse drug reaction monitoring centers the moment the National Coordination Center was with All India Institute of Medical Sciences at New Delhi. That moment, the efforts were also made to that India should also become the WHO collaborating center for pharmacovigilance program, WHO collaborating center under the WHO program for international drug monitoring. And it was in 1997 that the membership was floated, and we became the permanent member of the WHO program of international drug monitoring in the year 1998. And since then, you can see that the program, the quality systems are in place, the standard operating procedures are in place. We have expanded the program, and we are recommending the regulatory measures to the Central Drugs Standard Control Organization, and particularly the phase 4 of clinical trials, that is the post-marketing surveillance and the post-marketing trials, are very, very important part. And that's why the continuous safety monitoring of not only the medicines but also of the medical devices, the transfusion medicines, blood and blood related products is required, and pharmacovigilance program of India is taking all steps for monitoring the safety related aspect of all these items, and regularly sending the regulatory recommendations to the Central Drugs Standard Control Organization. We have identified 12 signals which are not reported earlier, and they are also not there in the prescribing information leaflet. The last signal which we have identified and confirmed is the paracetamol associated fixed drug reaction. And if we see such type of reactions from a very well established drug, then it makes us to think that the all drugs, they are not absolutely safe, and they require rigorous scrutiny, research, and checking up for safety and their risk benefit profile on continuous basis. As compared to the conventional drugs, however, the new drugs, they require more rigorous scrutiny in respect of the safety of medicines. Is anyone aware of what is the lifetime of a new drug? Any student? 5 years, 10 years, 12 years, 20 years. So I think you must take a message from today onwards and until further it undergoes changes, that the lifetime of a new drug is not the 20 years. It is the life of a patent from date of its application is 20 years, but the life of a new drug is how many years? 4 years, right? And for this, you have also to do the research, clinical research, like the post-marketing clinical trials you have to do, so as to detect, confirm, and identify the rare adverse drug reactions, right? And the Central Drugs Standard Control Organization requires the submission of periodic safety update reports for a period of 4 years, right? However, if they require that a particular drug requires more more data, or any moment if they require more data regarding the safety of medicines, the manufacturers have to mandatorily do it and submit it to the Central Drugs Standard Control Organization. And on voluntary basis, the adverse events, they should be submitted to the nearby ADR monitoring center under the pharmacovigilance program. In the context of the innovation and patents, the standards of the patents and proprietary medicines, they are set out in Schedule V of the Drugs and Cosmetics Act. So you can go through it. And in respect of the new drugs, you can in detail go through the new drugs and clinical trial rules 2019, and for conducting the clinical research, there are national ethical guidelines set out by the Indian Council of Medical Research. So, following a proper procedure in research, you can collect, collate the evidence and can publish in the scientific journals of repute and you can become a leader by considering 5 important things. There should be proper staffing, there should be proper planning, proper staffing, proper controlling, proper organizing, and then you can lead an organization. And a good leader is one before whom there are followers, and leader has to keep his team motivated and inspired, to keep the team dynamic, to bring out the innovations, to keep it forward, to take it forward. And all this entrepreneurship and running of business is also not free from the risk. So, by proper balancing the risk and the benefits, you can really take a lead in the area of pharmaceutical science, in the area of clinical research. And this place is a very good place for learning, and you can learn a lot by doing the experiment, by learning from your faculty and mentors. And I wish all the best to all of you. Thank you so much. If you have any question, I will be happy to reply to your questions. If there is no question, that means you are not attentive in listening. So listening is also a part of your research protocol. If you are not listening, then just hearing will not work, and you cannot innovate. Right? So listen properly, be attentive, and you can achieve your target with your hard work. And your hard work, no one can substitute with it, and you can succeed in your life. Thank you so much.
Thank you, sir, for your inspirational lecture. As your lecture was very focused on the student life, starting from their college, what do's and don'ts they do, and you were very interactive in with the students so that they can know what their mistakes are, and your knowledge about the pharmacovigilance was very is commendable. And people, the students, if they want to go for this pharmacovigilance career, it is really nice to listen to Dr. Jayaprakash sir. And you are really admired by the youth, the way you interact with the students, and the way you give your lectures. So I thanks once again for your presence. And now I would request Professor Dr. Bhupesh Sharma sir, along with Sandeep Arora sir, to felicitate our guest Dr. Jayaprakash Singh sir. Thank you, sir. Our next session is on clinical research methodology by Senior Manager Pharma by Dr. Himanshu Baveja, sir, Senior Manager Pharmalex India Private Limited. Dr. Baveja was previously pharmaceutical expert at WHO country office. He has done his master's degree in clinical research from Cranfield University, Bedford, UK, and many certificate courses in pharmacovigilance and clinical research. With over 15 years of experience in pharmaceutical, healthcare, and pharmacovigilance service industry, Dr. Himanshu brings a thorough understanding of pharmacovigilance process, the PV regulatory landscape, and the requirements. He supports the pharmacovigilance operation and training service areas in PharmaLex. His modus operandi includes combining domain expertise with strategic thinking for a holistic approach to business growth and implementation of transformational solutions along with new initiatives. He authored total 15 books on pharmacology, clinical research, pharmacovigilance for medical, pharmacy, and nursing students. He is also an associate editor and review board member of Indian Journal of Pharmacy and Pharmacovigilance. Thank you so much, sir, for your presence. And I would like to request you to please come on the dais so that the students can be benefited by your lecture.
Good evening, everyone. Thank you, Dr. Raman. Good evening, everyone. Thank you, Dr. Raman, for the kind words. And thanks to Dr. Sandeep Arora and the Vice Chancellor, Dr. Shukla, for inviting me in this conference, in the 5th International Conference. So let's have an interactive session on clinical research methodology. You are with me? Fine. So what do you understand by clinical research? Before starting the clinical research methodology, we just have to understand what is clinical research. So I want to know what is the basic understanding of yours on clinical research. What do you understand? Anyone, please? Yeah. Study? Okay. Fine. So, yeah, you are right. So any new drug, any investigational drug, we do a trial on the on the subjects. Subjects may be animals, may be human beings. Okay. When we do on the animals, that is called preclinical studies. And when we do on the humans, it comes as a phase 1, phase 2, phase 3, and phase 4. Right? Right or wrong? Fine. So as we do the clinical research on humans in phase 1, phase 2, phase 3, and phase 4, we have to design, right? We have to design our trial, that on which methodology we have to do the research. So in this, we will learn about the clinical research methodology, that what type of designs are there, how we implement that designs, and what are the modus of operandi in this particular clinical designs, clinical trials design. So clinical research is basically a type of research study which aims to evaluate the safety and effectiveness of the new medical treatments, devices, or the procedures. These trials are basically conducted on the human volunteers, as I already mentioned, phase 1, phase 2, phase 3, and phase 4. So after the phase 3, the new drug application is filed. I think all of you know this. New drug application is filed. Then we move to the phase 4. As Dr. Jayaprakash already mentioned, that in the phase 4 we do the pharmacovigilance. It is also called as post-marketing surveillance. So in the phase 4, as Dr. Jayaprakash very beautifully explained about the PVPI, that is a pharmacovigilance program of India, which is started in 2011, and he is one of the founder member. I know him since 2011 because I was also the part of PVPI at that particular time. So now I am moving to the clinical trials methodology. What is the importance of the clinical trial design? Okay. So clinical trial design is the important aspect of the interventional trials that serve to optimize and economize the clinical trial conduct. How we have to conduct a clinical trial, that depends on our design. What type of design we have to follow. The purpose of the clinical trial design is basically the assessment of the efficacy, safety, and the risk-benefit ratio. Every drug has a benefit, every drug has a risk-benefit ratio, every drug has a safety, every drug has efficacy. We have to compare our new drug, that is our investigational drug, with the standard drug. That is, a standard drug means well-established drug, which is already available in the market. Okay? यहाँ तक clear है? Is it clear? Fine. So goal may be a superiority, may be a non-inferiority or equivalence. the goal of conducting a clinical trial may be a superiority. We have to get a superior treatment for the patients, for the future patients, or maybe a non-inferiority, or maybe a equivalence. Equivalence means we have get the equal type of treatment for the future patients. A well-conducted study with a good design based on the robust hypothesis. Hypothesis is basically a research question. If anyone here is doing masters or PhD students are here, then they better understand hypothesis is basically a research question on which we work, on which we do the research. So hypothesis involved from the clinical practice and goes long way in facilitating the implementation of the best tenets of the evidence-based practice. A robust, well-powered trial adds to the meta-analysis, tribal evidence base, and contributes to the huge contributor of our knowledge medical practice. So based on our knowledge, based on our medical practice, we design the clinical trial, and this is the importance of the clinical trial design. Now, what are the uncontrolled clinical trials? As the name suggests, these are uncontrolled. So this design incorporates no control arm. We have two arms in the clinical trial. First is case, in which the investigational drug is given, and second arm is control arm, in which the already well-established drug, already standard drug of treatment is given to the patients. So as the name suggests, uncontrolled clinical trials. So in this, there is no control arm is there. The design is utilized to determine the pharmacokinetic properties of the drug. This design, the uncontrolled clinical trial, is basically used to determine the pharmacokinetic, that is a ADME. I hope all the students learn pharmacology. Pharmacokinetics is absorption, distribution, metabolism, and excretion. So in this particular uncontrolled clinical trials, we determine the pharmacokinetic property of the drug. Every drug has an ADME, right? So uncontrolled trials are known to produce a greater mean effect estimates than the control trial, thereby inflating the expectation from the intervention. We are conducting a clinical trial for some intervention that after a completion of clinical trial, we able to get a knowledge, we able to get some outcome from that particular exposure. Exposure we given to the patients, and outcome at the last we get from that particular clinical trial. So this is one of the uncontrolled clinical trial design. Now, what is the control arm options in the control trial? I already explained the uncontrolled trial. Now I am moving to the control trial. In the control trial, there is a control arm option is there. Now, as I already explained, there are two arms in the clinical trial. First is case arm, in which we give the investigational drug. Second is control arm, in which we give the standard treatment, right? So control trials allow the discrimination of the patient outcome from the outcome by the another factors such as the natural history, observer, or the patient expectation. So choosing a right control at the right dose and the right frequency is pivotal to the trial success. This line is very, very important, that choosing a right control at the right dose, right frequency is very, very important for the success of the trial. If we choose the correct and the optimized trial design, then the trial success is 100%. The control can be used as a placebo concurrent trial. Sometimes we use the placebo. Placebo means dummy drug. Dummy drug is given in the control arm. Okay. So this is a placebo concurrent trial. Placebo control helps to discriminate the outcome due to the intervention. Intervention means a new product from the outcome due to the another factors. Another factors may be a natural history, maybe observer, maybe a patient expectation, anything is there. But we have to understand that we are giving an exposure of the drug to both the arms and we are expecting an outcome. After outcome, we have to evaluate that outcome and then we come to the conclusion that this particular treatment of the case drug, that is our investigational drug, is better than the standard treatment or not. And what are the minimized side effects are there? Because every allopathic medicine has a side effects. I hope all of you agree with me, right? Yes or no? I know this is a long day, but I'm just trying to make the session interactive so that I don't allow anybody to sleep. I hope everyone is awake, right? Yes or no? Okay. So placebo control arm options in the control trials. So placebo must be used if there is no permanent harm, there is no permanent harm, death or irreversible morbidity occurs by delaying available active treatment for the duration of the trial and is preferable for the minimum risk and the short-term study. As I already mentioned, there are two groups, two arms are there. This is already explained in the diagram. That one is treatment group, as you already say. I'm just moving my cursor. This is a treatment group and this is a control group. In the treatment group, we are giving, as I already mentioned, investigational drug. And in the control group, we may give standard treatment or we may give placebo. It depends on the discretion of the sponsor. It depends on the discretion of the investigator. It depends on the discretion of the pharmaceutical company that what type of trial design they opt for their particular investigational drug. Okay, right or wrong? Fine. So target population is there. After target population, we random assignment is there to the two groups, that is experimental group and the control group. In the experimental group, we give the treatment, that is the investigational drug, and in the control group, we give the placebo, that is a dummy. And then we measure the outcome, that is a measurement of the outcome. That I already explained, and this is a diagrammatic picture of this particular explanation. Okay, clear? Please speak louder so that I also feel good. So design of the clinical trial design. So clinical trial designs are on two types, experimental studies and the observational studies. As I'm just moving to the next level in my slides, that two type of basic designs are there. I'm just not going in micro depth. On the overview, this is the experimental studies in which we give the experimental drug to the patients. In the observation study, we just observe the groups. We just observe, we just give the standard treatment and we just do the observational studies. In the experimental studies, there may be not controlled trial. It may be, as I already explained, not controlled, no control arm is there. Not randomized controlled trial, randomized controlled trial. There are the subtypes of the experimental studies. In the observational studies, these are the subtypes, that is maybe a case study. Case study as we give some particular drug to one patient and we want to publish that particular case in a literature as a research paper. So this is particular called as a case study. Case series, we give the particular drug to the 10 patients and that 10 patients we see the measurement of the outcome of that 10 patients, and that particular 10 patients we you can say we evaluate that 10 patients and we prepare the case series and that we publish the case series as a literature paper, as a research paper in our journal. Cross-sectional studies, descriptive cross-sectional study, I will go in detail in this presentation only. Ecological study is basically do on the population at the given point of time. We do the study of the specific population, any population. It may be a African population, it may be an Indian population, it may be any type of population is there. Cross-sectional analytical studies that I will go in detail in this presentation. Case control study and cohort study. Case control study, as I already explained in my previous slide, that two arms are there. One is case arm, second is control arm. And cohort study is given in the group of people. We select the group of people. For example, we select the diabetic patients as a group of people and we give that particular new drug, new investigational drug to the diabetic patient and see the outcome of that particular new investigational drug as a new diabetic drug. And then we came to the conclusion after the completion of our cohort study. Now, what are the clinical research designs? These are the clinical research designs. If there is no intervention, if we don't do any intervention, we just observe the patient, then it's called as observational study. In observational study, we do the comparison between the two groups. For example, we design the two group study in the observational study. Given we don't do any intervention, we don't give any new drug, we don't give any investigational drug, we just observe the patient's two groups and we just come to the conclusion that is yes or no. Yes means analytical study is there, we have to analyze, that is called analytical study, and then we can't able to analyze, then this will call descriptive study. If we do the intervention in the clinical trial, intervention means we are doing some new thing. Intervention means we are giving a new investigation drug. That is a experimental. Okay. So it is a random allocation is there. If there is a random allocation, then there are two options, yes or no. If yes is there, then there is a randomized control trial. And if no is there, then there is a non-randomized trial. We will go in detail in the next slide that what are randomized control trial and what are non-randomized trials. Randomized control trials in which we do the randomization of the patients. This is a type of the clinical trial design. We will explain in detail. Now, what is non-randomized trial? Non-randomized trials are defined as a trials where the investigator controls the allocation which is not at random. Allocation of the patient is controlled by the investigator in the non-randomized trial. Allocation of the patients is controlled by the investigator in the non-randomized trial. This is a type of the one of the subtype of the clinical trial design. In the control before and after the trials are defined, pre and post intervention outcome assessment and non-random group allocation is not under the control of the investigator. So if there is a control before and after the trials are defined by the pre and the post intervention outcome. There are two type of outcomes are there. Before giving the treatment and after giving the treatment. Before giving the treatment, that is called pre-intervention outcome, and after giving the treatment, that is a post-intervention outcome assessment. And this non-random group allocation is not under the control of the investigator. So in this, there is no allocation is not under the control of the investigator. Now, what are the randomized control trials? Non-randomized, I already explained. Now I am coming to the randomized control trial. Randomized control trials, the trial participants are randomly assigned to either treatment arm or the control. Two arms are there, as I already explained in the previous slide. One arm, investigational drug is given. Second arm, the standard treatment or the well-established treatment is given. So, in the randomized control trial, the trial participants are randomly assigned to either of the group, either of the case or either of the control arm. The process of randomly assigning the trial participants to the treatment of the control arm is called randomization. Now you understand that this is a method, this is a, you can say, a process of randomly assign a trial participant. Trial participants means patients which are participating in the particular clinical trial. Okay? Yes or no? Louder. So, process of randomly assigning a trial participants to the treatment or the control arm is called randomization. Different tools can be used to randomize control envelopes. These are the different methods by which we can do randomization. Closed envelopes are there, computer generated sequences are there, and the random numbers are there. Previously, we used the few years back, we used the closed envelope method. As of now, all the corporate hospitals, all the government hospitals use the computer generated sequence. Most of the, I am not saying all, most of the corporate hospitals and most of the sites, hospital investigation sites, use the computer generated sequence. So they generate the computer generated sequence on the random basis, and randomly they given without any bias. So this is a basis. This is a basically, you can say, a method to control the bias in the clinical trial. So this is a different tools, and in the randomized control trial, the trial participants are randomly assigned to the treatment and the control arm. So there is no bias is there. Nobody is biased, neither the investigator, nor the sponsor, nor the any person who is involved in the clinical research team. Now, these are the types of the randomized control trial. Simple randomization, as I already mentioned, that computer generated sequences are there in the simple randomization. Second is stratified randomization. In stratified randomization, this is a method of sampling in which we basically segregate the whole population into the specific subgroups based on the characteristics and based on the specific attributes and the characteristics. So this is called stratified randomization. Third is block randomization. Block randomization in which the two treatment blocks are well established, treatment 1 or treatment 2. These two blocks are basically designed so that we can do the randomization in the randomized control trial. So these are the advantages. Most probably most of the trials, you can see the website www.clinicaltrials.gov.in. You will get all the details of the clinical trials which are going on in India at any of the site of the in India and any of the hospital. So when you go to that site and see the clinical trials information, you see most of the trials are randomized control trials. Why? Because there are a lot of advantages of randomized control trial. First advantage is comparative. One treatment is directly compared to the another to establish the superiority. As I already mentioned, we have to establish the superiority between the investigational drug and the standard drug that is a well-established drug. So the one treatment is directly compared to the another to establish the superiority, minimize the bias, randomization, as I already mentioned, minimize the allocation bias and the selection bias, minimizing the confounding factor. So randomization makes the groups comparable according both known and the unknown factors. So we minimize the confounding factors and statistical reliability is there. When we do after the completion of the trials, we have to statistically analyze that particular trial. So when we do the statistical reliability, that statistical test of the significance is readily interpretable when study is randomized. So there are a lot of benefits of the randomized control trial. But as you know, there are two sides of the coin. So there are disadvantages also of the randomized control trial. So might demand the vast sample size which require more resources from the investigator. It requires very, very vast sample size, big sample size. Sometimes the allocation of the participants may be predictable, results in the selection bias. So sometimes it may be a predictable thing of the participants. Trial of longer duration and the more expensive. As the duration of the trial is basically, when the duration of the trial is long, then this directly proportionate to the expense, and that expense is beared by the sponsor company, the drug company, which is basically introducing the drug in the clinical trial. That is a marketing authorization holder. I hope everybody knows marketing authorization holder. So that cost is beared by the MAH, that is a marketing authorization holder, who wants to conduct that particular trial in that particular country. So trial of the longer duration are more expensive and results are always mimic the real life situations. Inclusion and exclusion criteria are there, highly controlled settings are there, but there are ethical limitations are also there. Some research we can't do ethically perform on the randomized control trial. So what is the parallel group design? Parallel group design, as the name suggests, two arms goes parallelly. One arm is treatment A, one arm is treatment B. So these are the type of the clinical trial design where these treatment and controls are allocated to the different individuals. Two arms are there, and which we will do the allocation and in the different individuals. So clinical trial in which each group of the patient receives the single treatment simultaneously, and parallel arm design is most used study design. So randomized and parallel, both are the mostly used study designs are there. Now this is the parallel group study design. This is a key element is randomization. As you already I think understood that randomization is one of the key step of, you can say, allocation, patient allocation during the clinical trial. So one treatment group and one treatment as usual, that is the standard group. Two active treatment groups are there. One of the treatment group receives the active comparator and another receives the standard treatment. Now crossover studies. Crossover studies are done for the improving the quality of life. There are a lot of drugs which don't treat the disease, they just improve the quality of life. Can you name some diseases? Vitamin C. Vitamin C is not a disease. Yeah. Okay, I simplify my question. My question is that the diseases which can't be cured, which can only be controlled, diabetes, blood pressure. So by this, I'm just taking the two examples for the in the interest of time: diabetes and the blood pressure, hypertension, I can say. So these two diseases, we can only control the disease. we can only improve the quality of life of the patient. We can't cure the disease. Diabetes can't be controlled, can't be cured, only controlled. Okay, either type 1 or type 2. Okay, hypertension, a same scenario. A patient have to take the antihypertensive for the whole life. So this crossover study is basically done for these particular type of trials. For example, diabetic trial, hypertension trials, and any another trial in which we just improve the quality of life. There are a lot of onco drugs are there, cancer drugs are there, which just improve the quality of life. They don't cure the patient completely. So clinical trials in which everyone considerably receives each of the treatment under the study. In this, everyone, all the patients, either the control or the case, they receive the treatment under the study. The study, each patient serves as its own control. Okay. So in these type of studies, each patient serves as its own control, and each patient gets the both the treatments. As I already mentioned, each patient gets the both the treatments. So as you see in the diagram, the randomized process, group 1, group 2, there is a medication A, medication B, and there is a washout period. And after the washout period, the patient who is taking medication A is given medication B, and patient who is taking medication B is given medication A. The washout period is a period in which we will don't give any treatment in the washout period, and that effect of the drug from the patient's body completely vanish, completely diminish. And after that, the treatment option will be changed. Now the variations of the crossover design. These are the variations of the crossover design, that the switchback and the multiple switchback designs are there, of the emerging relevance of the biosimilars. Biosimilars means after the patent expires, as Dr. Jayaprakash already mentioned, the generics will come into the market, and then we will do the crossover designs for some of the biosimilars. And now I'm moving to the next factorial design, that the studies involving 2 or more factors while randomizing are called factorial. So in this, we will given the 2 or more factors while randomizing are called factorial. Factorial design basically permits the researchers to investigate the joint effect of 2 or more factors on the dependable variable. In this factor, you can see in the diagram that sample population is there. The red you can see is a population, in the blue is a sample, and in this 4 arms are given. INT means investigational, and investigational A drug and the B drug. Investigational A and the PLBO means placebo, and the in second arm there is an investigational A drug and placebo B group. In third arm there is a placebo A and investigational B, and in the fourth there is a both the placebos are there, and we evaluate the outcome of all the 4 arms in the factorial design. This is also one of the design which is used in the clinical trial methodology in some of the treatments, in some of the design. Cohort studies are basically studies which are given, which are studied in the group of population. We, as I already explained, that is the diabetic patients are there, are basically powerful tool, basically very powerful tool in conducting a research in the human population. The population at the risk for the disease events is followed over a period of time. Any population who is a disease is followed over a period of time for the occurrence of the disease and the events, and the study used to estimate how the disease and the life events happened in this certain population. So these are the best method for determining the incidence and the natural history of the condition. So these are one of the best method in which we use this cohort study. This is a types of cohort study, very simple words, prospective and the retrospective. Prospective, we go in the presently, we do the study and see the result, see the outcome in the future. That is prospective. That is going forward. P means prospective, going forward. Retrospective means we have to move backward. That is, data is there at the hospital by the in the, you can say, in the HIS, that is the hospital information system. Okay, and we evaluate that data retrospectively. We reconstruct the outcome, we reconstruct the disease, and based on that we go in the retrospective, we move backward. We go in the past, we reconstruct the outcome, we reconstruct the disease, and we come to the conclusion. So these are the two designs in the cohort study, that is the prospective and the retrospective. Now the cross-sectional study is the type of observation study that is primarily used to determine the prevalence, that In the group of population, we see how the disease is prevalent in the given point of time in the particular population. Now, this is the target population and the sample size. Gather the data at the one time and both the exposure and the outcome. We see the exposure and the outcome present, exposed and the no outcome, unexposed and the outcome present, unexposed and the no outcome. All these things we will see in the cross-sectional study and these four by target population and study the sample size. Now, what is the case control study? I already explained. I will not go in detail. One arm is case, second arm is control, and this is the observational study. Maybe international study. We can do international also. We can do observation. Both type of studies we can do in the case control study. Now, nowadays, digital technology plays a very crucial and important role. This is very important slide. Digital technology has revolutionized the way clinical studies are conducted and providing the numerous benefits than the traditional method. पहले क्या होता था? Traditional method use होता था. Nowadays, digital technologies play a very important role in the clinical trials. How the digital technologies allows the real-time monitoring of the patient. As you know that during the COVID patient, the digital technologies play a very important role. It also plays a very important role in ethics committees meetings. It also plays a very important role in monitoring of the patient. It also helps the researcher in randomization. It not only saves the time and money, but also to ensure that the study products produce reliable results. The key benefit of the digital technology in the study design is the ability to reach to the wider audience. And with the online platforms and the mobile apps are there, the researchers can easily recruit the patients anywhere from the world, anywhere from the country. Now, the trial sponsors and the contract research organization have gradually increased the adoption of electronic informed consent and the EDC system to collect the electronic patient reported outcomes. So, this is the digital technology which is used, and in addition, the PRO instrument is replaced by the electronic versions. All the paper documents is replaced nowadays by the electronic version. I am just concluding my presentation. Rationale behind this clinical study design is basically, now I am coming to the conclusion. This is very important that the rationale जो होता है clinical study design का is to systematically investigate the medical intervention or the treatment or the hypothesis while minimizing the bias. The clinical trial design has to minimize the bias and ensure the reliable results. The study design determines how the data is collected, analyzed, and interpreted. It aims to answer the specific research question, establish the cause and effect relationship, and provide the evidence for the clinical decisions. So, different study designs such as the randomized control trials, observational studies, case control, all are based on the research objectives, ethical consideration, feasibility, and the potential limitations. So, these are the basically importance of the clinical trial design. These are my references. I am happy to take any question as of now, or if you can want to ask later, it's also okay with me. Any questions? Can I take the question?
Any questions, friends?
I'm happy to answer. The main aim to come here is just to share my knowledge with the youngsters. No questions? Can I take silence as a no question?
Any questions can be asked? You are discussing the method, methodology, the sources, and the required resources for the conduct of trial as well as entrepreneurship. In future, you can be entrepreneur, you have to think in that way. You don't have to sleep over the topic. Think in that way and you ask questions.
If in future you establish your CRO or SMO, CRO means Clinical Research Organization or Contract Research Organization, or SMO means Site Management Organization, then you have the sponsor comes to you with a clinical trial, that this particular clinical trial I have to conduct in India. For example, any company who is US based or UK based comes to you as a that we want to conduct this particular clinical trial. The sponsor will not tell you the clinical trial design. You have to judge, you have to see the drug, you have to see the patient outcome, you have to see the patient population, you have to see all the feasibility of the sites at the country level, and then you have to design the clinical trial. So clinical trial design, you have to be designed on your own, and you have to give the proposal to the sponsor. You want to ask him? Yeah, please. Yeah. Yeah. Good question. I will answer. Can it take 1 minute? Superiority means every drug, as I already mentioned, has a side effects, right? If the already well-established drug, for example, I take the example of oncology, cancer patient. There are a lot of cancer diseases are there which are just don't have any cure. They are only controlled by given a treatment. Okay, so a lot of pharma companies are there who are, you can say, working on the new drug development. Okay, so if any onco trial is there, we given a well-established drug for some blood cancer. We take the example of blood cancer. In the control arm, we give the well-established treatment for the blood cancer, and in the case arm, we give the new investigational drug. Right? New investigational drug, maybe XYZ is given. Okay, then we compare the outcome. Okay, superiority means that particular new drug is superior from the standard drug. Equivalence means there is no better, nothing is better. Both are more or less similar. Okay, non-inferiority means, for example, non-inferiority means the investigational drug is given to the onco patients, but their cost is very high. That is not affordable. For example, if I talk about India, India is not a developed country. India is a developing country. Lot of middle class is suffering from cancer nowadays. They can't afford the treatment of 15 lakhs, 25 lakhs, 50 lakhs. And that new drug, new treatment is cost is very high. Then there's no point of introducing that particular drug in that particular market. So that comes as a non-inferiority. I hope it's clear, or we will discuss after that in detail. Thank you very much.
One important thing that we have already highlighted in this discussion, that website clinicaltrial.gov.in. So if you are in workshop, the workshop continues even after you leave the hall. That should be the purpose of workshop. You should see as many cases, clinical trial registered, the processes, the methodology that you mentioned for individual drugs. The more you see, the more you will become expert. That is the purpose of the workshop. It is not just sitting here and going out. So kindly improve yourself. So mention, he has already mentioned. So as many cases you can see, that will increase your expertise. clinicaltrial.gov.in. And even for USA also, clinicaltrial.US website also, Indian website as well as US website. So the more cases you see, you get more expertise as well.
Thank you, sir, for your insights on a deep dive of clinical trials on the various terms, controlled and uncontrolled type, randomized trials. And you have summarized the clinical trials, which is in a form of a huge book chapter in a very simpler manner. Thank you so much. We'll not. As we are moving towards last session, so I would request, before that, I would request Dr. Nitin Sharma and Dr. Puneet Gupta to felicitate our speaker, Dr. Himanshu Baveja, along with Sandeep Arora, sir. So, to give a proper knowledge about clinical research resource management, we have our last speaker, Mr. Shuza Khan, Head Quality Assurance, PharmaLex Noida. Mr. Shuza Khan is a seasoned clinical research professional with 17 years of diverse global experience in leading quality assurance functions for clinical trials, early to late phase, and pharmacovigilance functions. Mr. Shuzar have held lead positions in reputed Indian multinational pharmaceutical companies like Torrent Pharmaceutical, Jubilant Pharma Limited, and clinical research organizations like Reptum Research Private Limited at PharmaLex India. He is a leading quality. He is leading the quality functions and is responsible for implementing a fully integrated quality assurance program and provide leadership throughout the organization in order to assure regulatory compliance. Mr. Khan holds Master's Honours degree in Organic Chemistry from Aligarh Muslim University, and he is certified ISO auditor and a GCP GCLP trainer, and is affiliated in for life with Indian Society for Clinical Research, and currently serving as a member for North Chapter. He's a speaker panelist at training conferences on GXP quality aspects of clinical research. I welcome you, sir, for your insights on the topic.
Okay, I think I am audible. Hi, good evening. Last session of the day. What a relief. So, thank you for all the kind words and having patience. Our profession is of patience. What to do? So we have chosen a patience for like profession of clinical research, and it have to be very patient because drug, as you already told, comes into the market and lifespan is very less, and it takes 15 years to come into a market and only 4 years to go from the market. So it's all the game about the patience. Now, what we will learn from this, I will just share my brief regard of the clinical research resource management. Now, what we have learned is we have already seen how clinical research is done. What is clinical research? Now we will let's now do a clinical research management. We have everything. Now we understood what clinical research is, what needs to be done to that. But what is the resources? How a clinical research will be done? This session will be focused on that part. So you will be learning what the key elements of resource management are and how you need to optimize or utilize these resources in conducting a clinical research. When we speak about leadership or entrepreneurship, then we think that everyone sitting in this room will set up a CRO or a pharmaceutical company. As Dr. Arora said, that should be the motive and mission. So whether we know how our resources will be managed, even in the university setup, if you see the university, you have a university setup, but what are the resources which are required and how you optimize these resources? So in any industry, whether it is clinical research or whether it is academic, it is about how you are managing your resources, which can optimize and how it can be cost effective, because at the end of day, the science is to bring a better healthier life to the society and also give the profits, because nothing is being done in the charity. Sorry to be very blunt, but when the industry or a sponsor or an MAH, what you say, if they are funding it, then they are thinking of charity also, benefit to the society also, and also getting the benefits from that, because it is a long process which they are going through and they are investing a huge amount. Then what's the end they are getting? Then new drugs cannot come until unless they are not investing in new drugs. So that is all about the clinical research resource management. Now, when we say clinical research resource management, as I already told you, it is this refers to efficient allocation and how we are managing. So now, where you will conduct a clinical research, there will be a requirement. Okay. So yeah, while he's setting up the technical glitches, let's continue. So what all research allocations are required for that? So one thing, can anyone tell for conducting a clinical research what all you need? Then guinea pigs we need a clinical trial office. So first thing, what I object, and now even with the new ICH GCP good clinical guidelines, we don't call subjects or volunteers. We call participants. They are participants in a research setup. They can be patients, they can be volunteers. So their participation can be voluntary, their participation can be on the healthy front also. So they are called the participants. So now we have the clinical research participants. We have a clinical research office, as we say. And what next? Drug investigator protocol. So all these are key resources, key elements. And who will conduct the study? Only investigator will conduct. So you need the staff, clinical coordinators, clinical research coordinators. So all these becomes the resources, right? So now we have to manage these resources. So that's why it is clinical resource management. It is not only you prepared a protocol, you have a funding, then what do you do about that? You don't have the qualified investigator, you don't have the resources to conduct, you don't have the sites, you don't have the ethics committee. Very important. Until unless it cannot be done, research cannot be conducted. Now you allocate these resources, we have all these parameters. Now we will utilize them. How we can utilize them? We will optimize them. The best way of the optimization to get properly channelized work, strategize. We need a strategy to optimize these resources so that our trials, clinical trials, can be conducted on time, right? So this is the way clinical research resource management is done. Now already Dr. Himanshu told you regarding clinical.gov. This is the recent data what is being displayed. Can I use mic? Oh, sorry. Can I? Thank you. This is much better. Okay. So now the number of clinical trials. Why clinical key resource management is important? Because you can see the data. You can see as on May 17, 2023, this is the current data. 4,52,604 trials have been registered globally. This is the global data of the clinical research. And already 64,000 are actively recruiting participants. Now, if you see the data of 2021, that was only 3,65,000, approx 1 lakh trials have increased globally. So, the number of clinical trials after the COVID, especially, we say before COVID era and after COVID era, as we say PC and AC. So, that's the way we categorize these eras. So, clinical trials, importance of the clinical trials, now everyone, general public also knows. So, our profession has been accepted by all. Earlier, no one was knowing what clinical research was, and COVID just what I can say educated everyone that what clinical trial is, and everyone now knows the importance of clinical trial. Then what we have, you can see the challenges. There's 50% wastage level of investigation product because we have take an example of any study crossover or whatsoever. 100 or 200 IPs we require and we ship from the manufacturer like 200 or 300 more IP. So the wastage has increased. What we do about that? Because our key management was not proper, we need to retain the IPs also because that was the regulatory requirement. Regulatory, when the audits, they take the IPs from the investigator site and they do analyze at their specific layer labs, specifically the Food and Drug Administration of US FDA, what we call. So we need to evaluate how this can be managed, and as a future entrepreneurs, we and you need to see that how clinical research can be best managed, because if there are so many challenges, then new drugs will never come into the market and it will take 15 to 20 years. Nowadays it hardly takes 10 years because we have computational chemistry, we have drug discoveries. Okay, so bioinformatics, chemoinformatics, you have everything. Technology is playing a big role. So that technology we have to leverage and see how we can leverage that technology to bring the drugs in the market best. Now, if already we have discussed that these are the key elements, personnel are required, facilities, equipments are required, funding, of course, without funding, clinical research cannot happen, time management. If a trial has to be set in 2 years, it cannot go for 5 years. So proper planning is required. Otherwise, it will be an endless job if time is not managed properly. Now, data. A lot of data is generated when a trial is conducted. So we need to manage those data. It is not only managing, it is analyzing also the data which is being generated. So we need to ensure that whatever the data is being managed or generated is being analyzed in a way that it is giving us the meaningful results or not. So that all becomes the key resources and evaluations of the resource management. Now let's have a just very quick breakup of all that. Personal management already we discussed. Now this is a part of human resource management, especially recruitment and hiring, training and development, compliance and regulation, continuous improvement. So if we do not have the skilled personnel, we do not have the experienced personnel, like university, we come across to the university, we do the campus, right, recruitments. So when we do these type of webinars, as Dr. Arora or Dr. Singh already said, that we are not looking for the evaluations, we are looking what is the caliber of these students, what university is developing. We coordinate with the universities so that industry also gains from them. It is not only about the like it is a benefit, two-way benefit. It is not only about the students getting the benefit, it is about the industry. Now the biggest challenge is the clinical research associates. We had recently in the month of Jan, we had the international science conference, and there it was discussed that the major problem we have with the development of clinical research associates, the industry is not getting the appropriate level of expertise what is being required. So how that can be done? Now many hospitals, many institutes like you as a MAT, once you do the MPharm and BPharm, you have postgraduate diplomas in clinical research, certification programs in clinical research, which is organized by different hospital setups of different clinical research organizations. So what we intend is to develop those courses in your MPharm itself, or BPharms or itself, so that universities also gain from it and we also gain it. So the importance of the skilled personnel comes here. So what we are doing by skilled personnel, we are ensuring patient safety, compliance with the regulation, quality data collection, protocol adherence, efficient study conduct. This is the main thing, and data analysis and interpretation. Now we have a lot of data. People do not know what to do with that. So what is the use of that data? Until unless the data generated is not analyzed properly. We have multiple softwares. We have SAS, we have other biostatistics statistics softwares, but the skilled persons are not there who do not know how to operate with these softwares. What is the use of these softwares? So we need to ensure that we know what we want to do with those data. So that is your personal effective management. Now, continuous improvement. We need to be updated and upgraded with the technology. We cannot remain 10 years what we used to do in the clinical trials or research. Now we have decentralized clinical trials. Because of short of time, Dr. Himanshu was not able to talk about that. Earlier it was patient used to come to the center. Now the research has changed. The research center is going to the patient. So that is your clear decentralized. I cannot go in detail about that, but that is the future. And India being the highest country where the clinical trials are again emerging. Earlier there was a very deep downfall of the clinical trial after 2012, but now after COVID, everyone, because we were not very efficient in our regulatory compliance. Now our regulatory compliance, which is CDSO, Central Drug Authority of India, has been very have changed their structure, and now the trials are being promoted with the current infrastructure. So now you can see the figure how 1 lakh trials have increased over the period of time of only 1 year. So that needs to be evaluated. Then again, already I have discussed this personal management. So personal management, we have researchers, principal investigators, study coordinators, data management, clinical research associate, regulatory affairs specialists, statisticians, and data analysts. These are the key roles of what we have defined. This presentation will be shared by you by Dr. Arora, so you can go deep down into that and evaluate that what all each role is responsible for. But to know these are the key roles for any personal management, what we call the personals, resources as a personal. Now we need a facility and equipment. This also play a very crucial role. Now what can be the facility and equipment? Clinical research center. You need a center to conduct the research. It can be hospital. It can be your phase 1 unit. It can be an hospital. Equipments. So how you analyze your research, like if it is a cardiac drug or it is hypersensitive, so you need to evaluate blood pressure and other critical factors. You need equipment from that. Pharmacy and drug storage, what you will do about the IPs, investigational product which you will receive. Storage is very important. If a shelf life is of a drug is 2 years or 6 years, you need to store in a way that drugs are not degraded. So that is very important. That right storage of a drug is very important. Then clinical trial management systems, as we already discussed, the softwares which we use. No one does the paper trials nowadays. Okay, so gone are the days when we used to write our findings. Even we have gadgets and tablets provided by the MHN sponsors to the every centers, and it is expected every centers to be fully equipped with them and to be trained on them. So as a clinical research personals, what we are sitting here, we need to be very acquainted with these softwares. So this plays a very important role in evaluating the data generated. There was a lot of frauds in India or across worlds in conducting clinical trials. Actual trials were not conducted. So with coming of the electronic systems, this fraudulent data collection has decreased. It has improved confidence on the data being generated in the trials, and ethical review facilities. Every research has to be ethically reviewed and approved. Until unless this cannot be conducted. So ethical research review is very important. Again, all those details have been provided. Funding, budget, planning, and oversight. Now you have a trial, everything you do have. You have all the setup. You have the strategy to conduct the trial, but you don't have the funding. What you will do? Nothing. So conducting a trial within the funding and budget planning is very important. MH is not giving any charity, as I already told. If a trial is conducted to be within a budget of 100 crores, then it has to be conducted within 100 crores. It cannot every day, it is not a like construction of a new house that every day your carpenter says this will all increase. So that cannot be happened in a scientific world. So proper planning has to be there to oversight your budget requirements. There can be multiple sources of fundings, grant applications, budget planning, all those aspects. There can be sponsors, there can be clinical trial cost, evaluations, transparency, disclosures, everything should be need to have an oversight of our budget planning. So sources already I have discussed. There can be government agencies, private investigators, charities, universities, research institutions, pharmaceutical companies, and budget variation. How what it will depend. The main things are study type and size, like phase 1, phase 2, phase 3. What is there? Location, multiple locations are there, or single location trial is there. What is the intended clinical budget? And site costs, startup fees, data collection, how much it will require, and patient care or procedures. So what we need is administrative, startup fees, time and sites, data, procedure, patient care, and personal. All these are very important to have evaluation of the budget and controlling of the oversight of the budgeting. Now, as I discussed time management, you need to have a proper time management. When we say personal, then personal will have to manage the time to conduct a trial in a more efficient way. How this can be done? We need to prioritize tasks. We need to have a calendars. There are multiple now Excel planners. We have Microsoft grants softwares where time management is done. So whenever a MH is being approached by any company, a proper planning is being shared where a Gantt charts are there. So in a stats, we need to evaluate that all proper time management has to be done, that what at what time period what will happen. So this is very important to conduct a clinical trial in an efficient manner, otherwise it will be an endless process. So these all aspects are very important to avoid any delays and bottlenecks. Data management, as I already informed, that you need to evaluate the data which is being generated, multi-center trials or single center trials. So all these aspects becomes very important. So this will include your multiple. Now everything comes around the personal. Now you will have data manager, database programmer, medical center or medical coder, clinical data coordinators, and others aspects. So all these are required to conduct proper data management. Among other key aspects for the resource management is regulatory compliance, collaboration, communication, risk management, technology and tools, training and development. So these all aspects are very important to conduct effective clinical trials, because at the end we need to ensure that clinical trials are conducted efficiently and with the regulatory compliance. Otherwise, we do the trials, everything is funded. What is the final aim? Final aim is to submit the dossier to the regulatory and approval. Until unless it is not approved, it will not go into the market. So what is the use? So regulatory compliance is very important. Again, all of this I have discussed, and I will just move ahead. Case study, I will ask Dr. Arora to give this a case study as an exercise to you. So this case study is just a brief, that this is a hypothetical case study for clinical research management, and where different challenges have been said, that this was a multiple phase 3 trial, and what were the challenges which was faced by the pharmaceutical company, and how these challenges need to be covered. So go through different aspects. There are multiple study material available on different channels. So please go through that. If you need to have a clinical research jobs in the clinical trials, or you need to open up your SMOs, site management organization, because this is the one of the most growing industry right now. I have seen many of my colleagues and juniors who have started their career in clinical research have opened their organizations. A simple clinical research organization, site management organization, which help in conduct of the trial. So you need to evaluate that, and this case study will help you. What are the challenges of this pharmaceutical company? How they come across the solutions and how they came across through the key effective management. So that's it from my end, and thank you, because everything I have already concluded that how important is that. So thank you and having beard with me. Thank you, doctor.
Thank you, sir, for adding little more into the clinical trials, because students have listened so much about clinical trials, but the resource management will be also required for conducting a clinical trial in a time-bound manner with the accurate results is also very important. And in healthcare, definitely the clinical trials are very important. So the way you have explained the clinical trials is really good, and I would request that the students will undergo this case study, and we will share the results with you so that so that we'll be aware that the students have understood the clinical trials in a proper manner. Now I would request Dr. Vikesh Shukla sir, along with Dr. Sandeep Arora sir, to felicitate our speaker, Shuza Khan sir.
Maybe my doctor has told you to take points. Secondly, entrepreneurship opportunity is already here discussed. To resource management also, there are many suppliers to the clinical trial area. What are those entrepreneurship possibilities? You have internet or your mobile, make a report. I have given a case.
Students, just 2 minutes, please. I would formally thank you for the patient listeners. Please, just 2 minutes more. I'll not take much time, please. So I stand here today as a proud and grateful organizer of the pre-conference workshop on clinical trials, innovation and IPR, entrepreneurship under the banner of ICEIL 2023. My thanks are due to our honorable founder president, sir, Dr. Ashok K. Chauhan, who have provided us the platform for conducting such intellectual events. My gratitude to our visionary chancellor, sir, Dr. Atul K. Chauhan, who is bringing new horizons to Amity Universe. My humble thanks to Professor Dr. Balvinder Shukla ma'am, our honorable Vice Chancellor ma'am, who have motivated us by her presence in this event and with her motivational words. I would like to extend my gratitude to our Dean, Professor Dr. B.C. Das sir, who have taken out his time. He has spared his time in spite of his not good health, as he told that he was not well since last 10 days, but still he was able to make out his time for this event. My appreciation to our HOE, Sir Dr. Arora, for arranging such events, because they give an opportunity to understand the subject beyond books by conducting such useful events. My gratitude to our Deputy Dean, Health and Allied Sciences, Dr. Tanveer Nawid, for always constant support and guidance. Finally, I would like to thank each and every one who are our guest of honor, Dr. J.P. Singh sir, Dr. Himanshu Baveja sir, Mr. Shuza Khan sir, for your valuable time and your insights on the topic in such a simple and easy manner. My thanks are also due to Mr. Naveen Dubey sir, who was not able to make out because of his pre-fixed business schedule, but he was able to join online. My thanks is also due to my colleagues who have given their valuable time. Dr. Bhupesh sir, Dr. Vikesh, Dr. Puneet Gupta, Dr. Nitin Sharma, Dr. Kalpana Nagpal, uh, who were being the patient listener along with the students. My thanks are also due to the student participants who have attended the workshop, and without you people, the event could not be a success. So my great thanks to you people for being a patient listener, and definitely these type of events will be helpful to you for understanding the topics better. My thanks to my co-coordinators, Dr. Rimpi Pava and Dr. Sweta Bavari, who were there for my support and making this event a grand success. And my thanks are also due to the student volunteers, a big team who were standing on toes since morning and yesterday to help in doing all the things to make the event successful. And last but not the least, yes, the IT team who is sitting behind the scenes, and the photography team who is capturing whole of the event. Thank you so much, everyone.